First of all, I'd like to thank all, Penny, and Oliver for giving me this great opportunity. I was a postdoc in Oliver's lab, and it is my great honor to present today. The title of my talk is Extracellular Tau as a Therapeutic Target of FTLD. Late onset neurodegenerative dementia are becoming a growing burden to society due to the gradual increase in life expectancy and dramatic rise in prevalence of these diseases. There are three types of neurodegenerative dementia: FTLD, Alzheimer's disease, and DLBD. Tau, amyloid beta, and alpha-synuclein are accumulated in each disease respectively. I talk about FTLD. First, I talk about iPS-tau, and second, talk about nasal vaccination. Corresponding to affected brain region, FTLD patients show behavioral disturbance, language impairment, and memory disturbance. Age of onset is 50 years old. Life expectancy after onset is 8.5 years. FTLD is characterized by prominent atrophy in the frontal and temporal lobes of the brain and tau accumulation. In a cell level, accumulating evidence strongly implicates misfolded proteins, especially oligomers, as a causative agent for most neurodegenerative dementia, including FTLD. Tau is axonal microtubule-associated protein that contributes to the stabilization of microtubules. Mutation in tau gene MAPT cause FTLD. There are 6 tau isoforms, the result of the splicing exon 2, 3, and 10. Alternative splicing of exon 10 encoding microtubule binding domain generates 3-repeat tau or whole repeat tau. Interestingly, both exonic and intonic mutations in MAPT cause FTLD. Tau intermediate and filament assembly were monitored by fluorescence to having S binding. Assembly kinetics of 3-repeat and whole repeat tau isoforms were assembled individually in the presence and absence of the reducing agent DTT. The assembly rate of whole repeat tau were greater than for 3-repeat, probably due to intramolecular disulfide bonds formed under oxidizing conditions. Transgenic mutant tau present oligomer tau in mice. We analyze oligomer tau in FTLD patient neurons. We have 2 research questions. Do oligomers present in FTLD patient neurons? Do oligomers play a pathological role in vivo? We generated iPS cells from FTLD patients with a mutation, intron, or exon. This patient presented FTLD. We established FTLD models using neurogen 2 induced direct neuronal differentiation from FTLD patient iPS cells with intron or exon mutation. This direct conversion method gave us a robust amount of cortical neurons. Generated neurons express messenger RNAs receptors of neurotransmitters. And electrophysiological analysis presented the functional properties to confirm whether iPS-derived neurons were functional. The electrophysiological property of iPS-derived neurons were evaluated by patch clamp analysis. Resting membrane potential of iPS-derived neurons were approximately -50. The intron mutation was corrected using CRISPR-Cas9. FTLD neurons with intron mutation showed increased whole lipid tau expression compared with lipid tau expression as previously reported. And correction of the mutation impairs the ratio in RNA level, also in protein level. FTLD neurons, both with intron mutation and exon mutation, have an accumulation of intracellular misfolded tau detected by immunohistochemical analysis using an anti-oligomeric aggregase antibody, TOC1 antibody. Some FTLD neurons exhibited typical misfolded tau puncta and dots, and control neurons, including gene-corrected line, were mostly negative for misfolded tau puncta or dots. Dot plot analysis presented accumulation of intracellular misfolded form of tau in non-denaturing condition using TOC1 antibody. We also analyzed misfolded tau by Western blot analysis using TOC1 antibody to detect tau species in denatured conditions. In FTLD neurons, usually with intron mutation or exon mutation, exhibited accumulation of tau species with higher molecular weight than control. Furthermore, FTLD neurons presented misfolded tau release into calcium medium as detected by dot plot analysis and confirmed by ELISA. Previously, it was reported that oligomeric tau disrupts intracellular calcium level and cause cell death. Misfolded tau exposure neurons exhibited increased neuronal death and intracellular calcium level. Thus, we evaluated calcium transits of FTLD tau neurons by calcium imaging assay. Electrical stimulation increased intracellular calcium level, and the increase ratio was greater in FTLD neurons compared with control neurons, indicating increased excitability in FTLD neurons. Next, we investigate vulnerability of FTLD neurons to degeneration and found they were more vulnerable to undergoing cell death than control neurons. We assessed mechanistic link between excitation of neurons and misfolded tau using DREADS. DREADS enabled us to control neural activity. We incorporated DREADS consisting of human muscarinic acetylcholine receptors bearing artificial mutations into FTLD iPS cells and generated neurons constitutively expressing DREADS. Expectedly, these designer neurons from iPS cells responded to CNO, M4D's exclusive agonist, and M3D's exclusive antagonist. Stimulation of M4D by its exclusive agonist, CNO. CNO inhibits calcium influx via GI signaling, decrease intracellular and extracellular misfolded tau, suggesting that neural activity may modulate tau pathology. Furthermore, inhibition of calcium influx by NMDA receptor inhibitor AP5 and AMPA receptor inhibitor CNQX increase survival of FTLD neurons. Collectively, the several phenotypes of FTLD neurons, including accumulation of misfolded tau and neuronal death, are similar for both intronic and exonic tau mutations, and neuronal activity relevant calcium influx modulate neurodegeneration in FTLD. This is a summary of first part of my talk. FTLD neurons exhibited accumulation and extracellular release of misfolded tau and neuronal death. These phenotypes are modulated by DREAD and glutamate receptor blockers. Our findings suggest that extracellular tau is a potential therapeutic target in FTLD. I talk about the second part. Tau is released from neurons into extracellular space, raising the importance of clearance extracellular tau by immunotherapy, including anti-tau vaccination. Several different lines of studies have demonstrated that efficacy of tau vaccination in FTLD mice. However, nasal vaccination has not been tested against FTLD, even though it offers many benefits over traditional vaccine approaches. We had 3 research questions. Can extracellular tau be a therapeutic target by nasal vaccination in FTLD? Does tau vaccination induce or inhibit glial inflammation? Can tau vaccination be monitored by tau PET imaging? So far, nasal vaccination has not been tested against FTLD, even though it offers many benefits. We developed nasal tau vaccination using Sendai virus vector. We constructed Sendai virus vector, which resulted in secreted tau being released into the extracellular space as antigen. Sendai virus is a murine parainfluenza virus type 1, negative sense single-stranded RNA, and has been used for the delivery of foreign genes to host cells. It has the potential to enter broad species and cell types by using salicylic acid on the host cell surface. The replication and transcription of Sendai virus vector occur and continue for about a month after administration in the cytoplasm of epithelial cell in the respiratory system, and its gene never integrate into host DNA. Sendai virus naturally replicates in respiratory mucosa, and according to a preferential route for Sendai virus vector administration in intranasal, this offers the advantage of induction of mucosal immune responses. Clinical trials using Sendai virus vector for gene therapy or vaccination have been conducted, and they show the safety and this treatment in humans. Sendai virus were administered to mice intranasally in our study, and the effect of the vaccination were evaluated. Efficient yield of an exogenous gene product in olfactory mucosa following nasal administration to mice were demonstrated with use of Sendai virus vector encoding EGFP. We investigate therapeutic effect of the vaccine in the murine model of FTLD through imaging and behavioral tasks. For tau vaccination, we constructed Sendai virus vector carrying cDNA of human tau isoform with 1N4R splicing with a P301S mutation as an immunogen fused to a signal peptide for directing gene products to a secretory pathway. Before vaccination to mice, we confirmed enhanced secretion of mutant tau into extracellular space by incorporating of this signal peptide by precipitating tau vaccine-infected cells culture medium. This led us detecting misfolded tau protein in the culture medium of tau vaccine-infected cells. In order to explore immunoreaction following tau vaccine inoculation, we analyzed anti-tau antibody generated in mouse serum. The production of antibody reactive to tau deposit in the hippocampus of FTLD mice was confirmed by treatment with serum from mice treated with tau vaccine relative to control vaccine. In addition, an increase of an anti-tau antibody in the brain of mice treated with tau vaccine was detected. We evaluated the effect of tau vaccine on FTLD mice using MRI and PET imaging. Volumetric analysis of MRI data demonstrated an elevation of hippocampus atrophy by tau vaccine, indicating that tau vaccine suppressed neural loss. These FTLD mice were also subjected to PET scan to assess the time course of the neuroinflammatory changes. In vivo PET imaging with carbon-11 AC5216 radioligand for TSPO, a marker for detrimental gliosis, indicated that FTLD mice with control vaccine began to show an elevated level of TSPO in hippocampus at 6 months age, and this became even more pronounced at 9 months. But vaccine treatment inhibited glial inflammation in FTLD mice. This relatively late change in TSPO PET was attributable to robust microgliosis, as has been previously documented, and was notably suppressed by tau vaccine treatment. However, a modest but insignificant rise of TSPO PET signal in the hippocampus was transiently observed at 3 weeks after immunization with tau vaccine. Next, we check tau pathology. The decreased deposition of phosphorylated tau in hippocampus CSO neurons of FTLD mice by tau vaccine was further proven by immunohistochemical analysis. Postmortem mouse brain tissue collected as a predetermined endpoint of the therapeutic evaluation at 9 months of age. Finally, we tested cognitive impairment of FTLD tau mice. Control vaccine-treated FTLD mice exhibited a loss of hazard perception in the elevated pulse maze test and disturbed spatial memory in the Burns maze test, but both were significantly alleviated by treatment with tau vaccine. Impairments of contextual memory and hazard perception as monitored by fair conditioning tests were also observed in control vaccine-treated FTLD mice, but again, that was ameliorated by the tau vaccine vaccination. These cognitive impairment phenotypes are known to be associated with hippocampus and other limbic area, reinforcing the notion that tau vaccine served to rescue hippocampal neuron from tau-trigger dysfunction. Other behavioral scores such as performance in the open field, social interaction, not-out treadmill, operate, preparation inhibition, and trial suspension tests were not altered in FTLD mice related to control mice, irrespective of treatment. Taken together, these findings provide evidence for the depression of tau-induced cognitive deterioration in vaccinated FTLD mice. A PET radioligand for fibrillary tau inclusion, carbon-11 PBB3, is capable of visualizing densely packed tau alleles in the brainstem of FTLD mice, but carbon-11 PBB3 PET does not detect neurotoxic pre-tangle to measure tau pathology in the hippocampus of these animals. In our study, there was a trend to suppress carbon-11 PBB3 positive tau deposition in the FTLD mice brainstem at 24 weeks after tau vaccine injection, but the difference in PET signal between the treatment groups was not statistically significant. This is a summary of the second part of my talk. Extracellular tau is a potential therapeutic target in FTLD. So maybe a different types of glial inflammation, acute inflammation by vaccination, and chronic inflammation by neurodegeneration. Tau PET imaging didn't monitor the efficacy of tau vaccination in FTLD mice. Gene therapy-based vaccination may be a therapeutic option for tauopathies, including Alzheimer's disease. I'd like to thank all collaborators, co-workers. Thank you for your attention. Thank you.